Hi everyone, this is Georgia and I'll be presenting the second half of the article, which includes figures 4, 5, and our proposed experiment. First, we'll take a look at figure 4, which is about trying to isolate the beta cells from the other cell types, because there's currently a heterogeneous mixture of alpha, beta, EC, and other non-endocrine cells. Before we jump into panel A, just a quick reminder about how we read these figures. Each point on the figure is an individual cell because they did single-cell RNA sequencing, and the closer the points are together, the more similar the transcriptomes of the cells were. So that means that cells that are close together are very similar, and cells that are far apart are really different. So for example, here we see that the beta cells and the EC cells are close together, but the non-endocrine cells are all the way over here, separated from the endocrine cells. So in panel A, the authors figured out, based on their whole transcriptome analysis, that CD49A is a surface marker for the beta cells. And this is apparent here, where the expression in the beta cells is 10 to 100 times greater than the alpha and EC cells. And that's why they chose to use CD49A to purify the beta cells. This is panel B of the same figure. First, they separated out the non-endocrine cells by separating all of the cells and passing them through this mesh filter. And then the endocrine cells reaggregated on their own, but the progenitor cells stayed in the supername where they were then discarded. And the reaggregated cells are in the center column of the figure, and the first column has the original cells. So then the cells were stained with anti-CD49A antibodies conjugated with PE, and then microbeads that had anti-PE were added, so the CD49A positive cells were attached to the microbeads. So pretty much the CD49A surface marker attaches to its antibody, which is also attached to PE, which then binds to the anti-PE antibody on the microbead. And the cells are separated magnetically, and the column on the right is the cells after this magnetic sorting. The top row shows the markers for only the beta cells, and the bottom row shows the markers for the beta cells, alpha cells, and EC cells, so all of the endocrine cells. And the takeaway from this figure is that after sorting, the beta cells are concentrated, which is why the cluster in the top right here is so bright, but the native and reaggregated clusters kind of look the same. And one limitation of this approach is that some other cell types also express CD49A, but at lower levels than the beta cells. In panel C, the authors did flow cytometry to determine what percentage of the cells were beta cells or EC cells. They used C-peptide and NKX6.1 to identify the beta cells, and SLC18A1 to identify the EC cells. And again, they measured the cells at the beginning, after reaggregation, and after sorting. In the first part of the figure, you can see that before sorting, around 50% of the cells had beta cell markers, but after sorting, more than 80% of the cells had the markers. And on the other hand, before sorting, around 25% of the cells had the EC markers, but after sorting, less than 5% did. This shows that the purification process was fairly successful. In panel D, the next step was to determine the functionality of the beta cells in response to glucose. This is the same glucose sensitivity test that we talked about before, where the cells were switched between the low glucose media and the high glucose media, and the stimulation index is that ratio of insulin secretion in the high glucose environment to the low glucose environment. And the insulin secretion was measured with an ELISA assay. This figure shows that the cells were around more or less 5 times more responsive to glucose after sorting, which makes sense because the beta cells were purified. The insulin levels were normalized to the total number of cells, so in the native and reaggregated groups, the responsiveness is kind of diluted by non-beta cells. So there's like beta cells and non-beta cells in there, which made them less responsive. Now we'll move on to figure 5, which is pretty much where the authors developed their model for endocrine cell differentiation in islets. And we're going to skim over the first lots of parts of the figure pretty fast, but I wanted to give you guys the key points so you know how the authors reach the conclusions that they come to at the end. And their overall goal was to decide if the beta cells and the EC cells developed separately and ended up having similar transcriptomes because they kind of differentiated in parallel paths, or if they developed from each other in a sequential fashion. And this wasn't immediately obvious to them, because both the beta and the EC cells appeared during stage 5 of the experiment, so it was hard to tell which one came first or if they appeared at the same time. So to resolve this question, they sequenced the cells at different days throughout phase 5, and panel A is just showing, in that same type of figure as before, the days when the cells were sampled, and panel B shows the expression levels of neuro-G3, which is a regulator of endocrine induction. And again, these figures came from the RNA sequencing. So we're kind of going to gloss over these two, but panel C shows how the authors categorized the cell types based on their transcriptomes. And they pretty much identified two branches in the differentiation, where NKX6.1 progenitors split into non-endocrine cells and early endocrine induction cells, and then the late endocrine induction cells split into beta cells and EC cells. Panel D just shows the fraction of each cell type on each day of the experiment, and it shows a general trend from progenitor cells to differentiated cells. We're also going to skip through E and F, but the purpose of this part of the study was to confirm the branching hypothesis of the late endocrine induction cells to the beta cells and EC cells. The actual math here is kind of complicated, but in panel E they assign these pseudo-time values to the cells, and in panel F they show how the beta cell markers and EC cell markers fluctuate based on pseudo-time. We're going to skip over G as well, but pretty much they just made a math model using pseudo-time and the assigned branches covariates, and they used fancy statistics to compare the branched model to an unbranched model, and they ended up identifying 313 genes that were significantly associated with a branch, and a lot of these were transcription factors and gene-specific things to beta cells and EC cells. Okay, so here's the important part. H is kind of small because it just shows the data that supports the rest of this, but it's a map that shows how the authors used relative gene expression for a number of different genes to differentiate between the various cell types. Panel I is the take-home message, which is the proposed differentiation pathway for endocrine induction. The first split is right away from the PDX1 progenitor cells to endocrine and exocrine cells, and even the pathway that leads to alpha cells splits off right at the beginning. And for the cells destined for the endocrine fate, the NKX6.1 progenitors then lead directly through early, middle, and late endocrine induction, and the last step is to split into the beta cells and the EC cells. Now, some limitations of this study are that the cell sorting markers that they used are also expressed by some other cell types, and the purification process wasn't perfect, but it was pretty good since they got around 80% beta cells. Another limitation that was mentioned in the paper is that the transcriptome of the stem cell-derived beta cells differs in some ways from the transcriptome of the native beta cells. And this is going to be the topic for a proposed experiment a little bit later. Some future applications from this study is that the proposed development pathway we talked about at the end can be used to develop a more efficient differentiation protocol for beta cells. A long-term application of this work is going to be able to grow beta cells in vitro to transplant them into patients with diabetes. We can also use this technology to model islet cell disease and to test various treatment methods in trials. Okay, so to switch gears, here's the basis for our proposed experiment. The authors mentioned near the end of the paper that the stem cell-derived beta cells differ in gene expression from the bona fide beta cells, and they also said the transcriptome might change to match better if the cells are implanted into a live animal. So that's the hypothesis that we're going to test. The question we're trying to answer is, will the beta cells be able to adjust to match the transcriptional profiles of natural beta cells after they're transplanted into an animal? We hypothesized that if the beta cells are implanted subcutaneously into either a healthy or a diabetic mouse, they'll respond to the environment of the mouse and their transcriptional profile will change to match the transcriptional profile of the natural beta cells. So just a quick refresher on diabetes, because the purpose of this experiment is to develop a treatment option for diabetes. In a healthy pancreas, the beta cells secrete insulin in response to glucose, which then tells the cells in the rest of the body to take up glucose. And type 1 diabetes results from an autoimmune attack on the beta cells, so they can't secrete insulin, so there's no insulin telling the cells to take up glucose. And type 2 diabetes is a bit different because the beta cells are secreting insulin, but the rest of the cells don't respond to it. Islet transplantation has been proposed as a possible treatment for type 1 diabetes, and there's been a few studies to support this. but that would only work for type 1 because you're adding beta cells that can secrete insulin, but it wouldn't fix the problem that's happening with type 2 diabetes. So here's the methods for the proposed experiment. First, we would differentiate the human pluripotent stem cells into the islet cells using the same protocol that they used in the article. Without going through the whole thing again, we're going to grow them in the differentiation flasks with the growth factors and other small molecules to promote islet differentiation. Then we'll isolate the beta cells using the CD49A beta cell surface marker. So again, we'll mark them with the anti-CD49 antibody with PE and stick them on the magnetic microbeads with the anti-PE, and then sort them magnetically. Then we'll implant the cells subcutaneously into both healthy and diabetic mice. We're including the diabetic mice to see if they can regain the function that the beta cells are supposed to be doing. And for a qualitative analysis, we'll use RNA sequencing to compare the transcriptome of the implanted beta cells with the beta cells from both healthy and diabetic controls, as well as the beta cells differentiated in vitro. So we're comparing a few groups here: the stem cell-derived beta cells grown in vitro, which are the ones from the original study, the ones grown in both healthy and diabetic mice, and the bona fide beta cells from healthy and diabetic controls. We'll also do a quantitative analysis of beta cell function with the glucose sensitivity assay, and the cells, again, will be switched between the high glucose and low glucose media, and the insulin production will be measured with an ELISA assay. Again, the glucose sensitivity index is the ratio of the high glucose insulin levels to the low glucose insulin levels. The expected results of this study are that the implanted beta cells will have a transcriptome that matches the bona fide beta cells more closely than the stem cell-derived beta cells in vitro. This is because there are so many factors in an animal model that can influence gene expression, and you can't control all of them in a lab. However, there wouldn't be any expected differences between the cells implanted into diabetic and healthy mice, because the beta cells should function independently from the cells in the pancreas because they were implanted subcutaneously, so they should work properly on their own. We'd also expect that the cells grown in the mouse will have a higher glucose stimulation index than those grown in vitro, but probably lower than the bona fide cells from the healthy individual. Hopefully, they'd have a much higher stimulation index than the diabetic control, which would provide support for this as a treatment option after further studies. There are a few limitations to our proposed experiment, and the first is that we used immunocompromised mice, which we had to do because we were putting human cells in them. As technology stands, when islet cell transplants are done in clinic, they're done with immunosuppression, but if this could be avoided, that would be ideal because immunosuppression can create a lot of complications for patients. Another way to avoid this would be to encapsulate the cells with some sort of polymer so they don't set off the immune system, and there's another group working on that. Stem cells could also be taken directly from the patient, but that would be expensive and time-consuming. Another limitation is, as I mentioned before, this technique wouldn't be useful for type 2 diabetes because that's a different problem physiologically. A future direction from this could be to identify genes that are being undertranscribed in the beta cells that aren't working, and try to use some sort of gene therapy to upregulate them. So here's our sources from this presentation. Thank you all for listening. I hope you enjoyed our presentation and maybe got some clarification about some of the figures in the article. Let us know what you think in the comments.